Let's talk about multifactorial inheritance and common diseases. Multifactorial inheritance refers to the fact that many factors make sure inherit a specific trait, phenotype, or disease. So this is due to multiple genes and multiple environmental factors. And common diseases refers to the fact that we have specific diseases which are very common, and common diseases are specifically genetic variants found in 1 to 5% of the population. And examples of this include type 2 diabetes, asthma, and these are very common, and these are due to many factors. So these are multifactorial inheritance. Now I want you to understand the difference between monogenic, polygenic, and multifactorial inheritance. So monogenic refers to a single gene mutation determines a trait. So examples of this are autosomal recessive cystic fibrosis or sickle cell anemia. Monogenic inheritance typically are much more rare than multifactorial inheritance, although not always, right? Because multifactorial inheritance explains these common diseases. And polygenic inheritance refers to multiple genes contributing to a single trait, and multifactorial inheritance refers to the fact that many genes interact with each other and environmental factors, and these all together express and are responsible for a single trait or disease. Examples of this include type 2 diabetes, asthma, schizophrenia, high blood pressure, heart disease, some types of cancer, lung cancer, so on and so forth. Now for multifactorial inheritance, we work with the multifactorial basic model, which explains quantitative traits such as blood pressure and height. And this is a bell-shaped distribution curve, as you guys see here, explaining that many of these quantitative multifactorial inheritance traits, quantitative multifactorial traits can be explained via this bell-shaped curve. So let's take an example here with height, right? If you're homozygous recessive, you're shorter than if you were homozygous dominant. And this is due to that every time you have a dominant allele, you will add a little bit of height, right? You could add, let's say, 2 or 3, 4 centimeters. Now, probably more realistic is the one you're seeing here. That height is determined by 2 loci instead of 1. And the second loci also has 2 alleles. So capital B for the tall one and little b for the short one. And here we see 9 possible genotypes, giving us 5 distinct phenotypes, right? And here you're homozygous recessive, here you're homozygous dominant. Every time you have a dominant allele, you will add a little bit of height. You will add, let's say, 2 centimeters of height to your baseline, which could be 145 centimeters, right? And so the height is the sum of all the tall alleles. And by the way, remember that every loci follows Mendelian law. Now, this does not yet look like this bell-shaped curve, but we can now extend this example so that it includes more genes and also include a bunch of environmental factors influencing height. And then you'll actually see that you'll have this bell-shaped curve. Another model is the threshold model, which explains that a disease could either be absent or present. So this is a all or nothing kind of model. And once you've reached the threshold, well, now you have the liability combination of the mother and father for both genetic and non-genetic environmental factors. And you'll get the specific trait, phenotype, or disease. So this is the threshold model for all or nothing phenotypes or traits. Now, of course, the recurrence risk in monogenic and multifactorial diseases is different. And monogenic diseases, as you guys know, the recurrence risk is 50% for autosomal dominant and 25% for autosomal recessive. Now, for multifactorial diseases, the occurrence risk is a bit more complicated. And here we utilize empirical estimates based on a whole bunch of observations. And here we can also utilize twin studies and things of that sort, right? Specifically zygotic and monozygotic and dizygotic twins, so also known as fraternal and identical twins, to kind of understand multifactorial diseases better, understand the complex interaction between the genes and environment, because sometimes the genes play a bigger role than the environment, and sometimes otherwise. So you can use these twin studies to say something about specific types of multifactorial diseases. Now let's talk a little bit about genome-wide association studies. And genome-wide association studies is a systematic investigation of the genetic material, often 1 million markers, in which one searches for variants that are more common in some group cases than other controls. And in genome-wide association studies, you're identifying weak risk alleles within multifactorial diseases. And this is specifically used to find the genetic variations, alleles in the genome that occur more frequently among cases and controls. So basically what you're doing here is you're identifying gene loci for a specific trait or phenotype. You're kind of examining and looking like at the whole genome to identify SNPs and associating these SNPs with a specific trait or phenotype. Now the problem with this is that just because you've associated SNPs with a specific trait or phenotype does not mean that SNP is the causation of that phenotype or trait, right? The genes associated with the trait does not necessarily mean causation, right? Remember, because we're talking about multifactorial inheritance, which refers to that many and a bunch of factors influence inheritance. So sometimes you could say that GWAS makes it so that we have a lack of utility, or a lack of understanding of if the SNPs can actually be useful to say something about this specific trait. The good thing with genome-wide association studies is you don't need to know anything beforehand. These are hypothesis 3. So basically, you can come with new biological pathways not previously suspected or found out of nothing. So in that case, SNPs can be, GWAS can be very, very good to just learn new things about these multifactorial inheritance traits or diseases. But again, we have this problem with that, the fact that the SNPs might cause disease, or it might be in linkage disequilibrium with a nearby variant causing the disease. So we need to be careful with this, with just interpreting a genome-wide association studies, and we need to be very cautious when interpreting this, because various factors can produce associations between a disease and a potential risk factor. Also, there's some other problems with genome-wide association studies. For example, that some diseases or traits are more common in some parts of the population than others. For example, type 2 diabetes is more common in specific ethnic groups in comparison to other ethnic groups. So for example, in the Pima Native American population, the type 2 diabetes is much more common, relatively speaking, to the European population. And that is why we need to be very, very cautious when interpreting genome-wide association studies.